Thanks, Bob. And actually, several of these students and postdocs are in the audience, and I'll mention some of them as we go through this material. But my goal here is to try to wrap a lot of this together and look at how can we apply this in a breeding program, and what are the challenges, and not specific to PRRS, but disease in general, host response to this infectious disease. And quite a bit of the material that I'll show actually is also in the online course in a slightly different form. Okay, so genetics has been very successful, and those of you who listened to Allison's talk yesterday, she gave some very good illustrations of the success of quantitative genetics and animal breeding, which up to recently really has been a black box approach. We know that the traits we're interested in, so growth rate, reproduction, litter size, and so on, that they're affected by genes, but we really, up to recently, we really didn't utilize any information on where those genes are or even how many genes there were. We didn't know how many genes there were that controlled growth rate or where they were. So from that perspective, it was a black box. All we knew that the phenotype that we have is heritable, and that basically says, well, what proportion of the differences we see in phenotype is due to the genetics of the pig versus environment. So, but we've been in animal breeding, we've been very effective in using statistics to utilize all that information of the phenotypes on the individual that we measure on the individual itself and also its relatives to estimate breeding values, to estimate the genetic value of an animal as a parent, and then utilize that in selection programs. And I'll go through this fairly quickly because basically what Allison illustrated yesterday also is that that has resulted in remarkable advances in pig production. We can now produce a pig that we take them to larger market weights with on less feed than we did 40 years ago. It's actually a slide from Graham. Same in dairy cattle. We have seen tremendous increases in milk production, which has resulted in us being able to, as a country, be able to produce more milk with much fewer cows, and therefore leading to much lower impact on the carbon footprint. And in chickens, and again, this is from University of Alberta, showing broilers from 57, and in 2005, after a 42-day growth period. The tremendous advances. So it's been very successful for many traits, but not for health. We continue to see lots of infectious disease in not just pigs, but all livestock. And genetic selection has really struggled to improve pigs in terms of their health and their ability to resist infection. And that's, of course, why we embarked on this large project of the genetics of PRRS and other diseases. And, of course, there's future diseases on the horizon, African swine fever, and foot and mouth, and so we'll need to prepare ourselves in terms of the genetics of the pig to be ready for future diseases that may be future pathogens that may come into the U.S. and North America. Now, if you look at resistance to infectious pathogens, there are some pathogens where the host can control that pathogen by a single gene, by a mutation in a single gene. And the F18 E. coli is a good example where there's a mutation in the receptor that the E. coli F18 bacteria uses that allows the bacteria not to bind to the receptor. And people have shown that provides complete resistance to F18 or E. coli. And in other species, there are other examples of single genes that give complete resistance. And there's K88 E. coli is another example in pigs. So there are some pathogens that are controlled by a single gene, and we now also know that in PRRS we can create a change in a single gene that makes them completely resistant. The difference with this mutation compared to F18 E. coli mutation, the FAT1 mutation, is that this is not naturally occurring. Okay, in all the pigs that we've looked at, that Bob's looked at, from commercial pigs, there's none that show no viremia after infection. So complete resistance doesn't seem to be occurring in terms of the natural variation that we see among animals. So but there is partial resistance as we've found and what we've seen. Okay, but so if you get away from the gene editing, if you look at the natural resistance to PRRS, it's incomplete and it's polygenic, which means that it's affected by many genes. So this is the GWAS, this Manhattan plot that Bob also talked about, where we did find in chromosome 4, the chromosome 4 mutation, the one telephone pole in a wheat field. Right? And so, yeah, there are some genes that explain a sizable amount of the genetic differences between pigs in resistance. It explains about 15% of the genetic variance. And there's some other regions, but they explain together, even if you want to add up the ones that are significant here, it doesn't go over 20%. So there's still 80% of the genetic variation in host response that we can't find, that is in the wheat field. Right? And it's everybody's guess, which of these dots they are, because there could be lots of false negatives, false positives. So that's a challenge. Right? And so there's no single bullet. If you stay away from gene editing, there's no single bullet solution to improving the genetics of PRRS. Now, we do have a marker that's available, and so one of the decisions we made as a group is that we wouldn't attach any intellectual property to this marker, but it's freely available for anybody to use. The other question, of course, is resistance, tolerance, or resilience, and Andrea talked about that. So, so resistance has a very polygenic basis, apart maybe from WUR, but then other genes, many genes with small effects. Tolerance probably has a very polygenic basis, many genes involved, and the same with resilience. Okay. So that's a challenge, and this is, if you look at the GWAS for weight gain under infection from the PHGC trials, again you see the one telephone pole, but the rest is, is wheat, right? So there's lots of genes there that there's almost 90% of the genetic variation is down here, and they represent many genes with small effects. And if you think of the biology of growth, even under infection, I mean, it's not surprising that there's many genes that are involved in creating genetic differences, that are involved in controlling growth rate. So that does pose a lot of big challenge. So how do we then go about genetically selecting animals that are better able to resist or be resilient or tolerant to these quantitative or complex disease traits? And in livestock, we're not the only ones. I mean, in human genetics, when you look at obesity, there are a couple of genes that have a substantial effect that increase risk of getting obesity, but there's lots of genes with small effects. And the same with other diseases, cancer, for example. So the challenge for working with, so there's not a single marker that we can select for. We can select on WR, and that does some good, but it still leaves over 85% of the genetic variation untouched. And it comes back to, the challenge comes back to the breeding pyramid that Graham also talked about, that the nucleus lines where all the selection takes place, they have to be maintained very biosecure. Okay, and so we're not able to measure disease resistance, there's very limited disease there. So the expression of resistance and tolerance to disease we cannot observe on the selection animals. Okay, so it's very difficult to obtain relevant phenotypic records to do the same thing that we do for growth rate. You know, we can measure growth rate here very easily and select on that, but we cannot do that for disease traits because of the high health nucleus farms that they're on. Also, even if we do measure, if we measure disease traits here, they typically have low heritability. So there's a lot of noise when you look at the disease diagnostics. Just a diagnostic test has false positives, false negatives. But whether or not an animal gets infected, there's a lot of environmental factors that play a role, even between animals within the same barn. So when we estimate heritabilities of field data on disease, they typically have very low heritability. Okay, that doesn't mean that there's no genetics involved. It just gets overwhelmed with environmental variation, and we can't see the forest through the trees. And then always the question is, well, yeah, should you focus on selecting just for one disease, right? And the danger that if you select for improved resistance to one disease, it may be counterproductive for other diseases. And, and that's why the study that Graham talked about is so crucial, that we look at a complement of diseases and find animals that are resilient to a battery of diseases. Okay, so, but even if we, could collect some of these phenotypes, now how will we select for that? Well, okay, so these are some possible things we could do. We could select, we could measure indicator traits in the nucleus, and Graham talked a little bit about that, things that we can measure in his the study that he talked about, that are measured on young animals while they're still in the healthy environment before challenge. We could also, we could go out and measure phenotypes at the commercial level, okay? Or we could collect phenotypes in special experimental facilities and then utilize that information in the nucleus. And I'll talk about all three of those approaches and some examples. Okay, and then we have possible selection criteria. We could just use it just like we would do with growth data. We put it through our statistical machinery, and we estimate breeding values for disease resilience of the nucleus animals and incorporate that in selection index. We could, if we have some major genes or genetic markers like WR, we could, if we've been able to pull some genes out of this black box, we could directly select on those. We can genotype the animals for those markers and select on that, and it can already be done for WR. Or we use an approach that's called Genomic Prediction, and I'll explain later on what that is. It basically involves using not just a small number of markers, but using a large number of markers across the genome. So those are the selection approaches. So some indicator traits. So one thought would be, well, a lot of individual or a lot of farms, they vaccinate for PRRS with a modified live virus. Well, maybe we can use response to the modified live virus vaccination as an indicator trait. Now, we still wouldn't be able to measure that in the nucleus, because you don't want to use a modified live virus vaccine in a nucleus on the breeding animals, but we could go out and measure that on a number of farms. You control when you infect them with the modified live virus, and then you could measure response. And we actually did that in some of the PHGC trials, where we had what Bob and Joan talked about, the vaccination co-infection trials. This shows one of those trials, the PRRS viremia of the animals after co-infection of the non-vaccinated animals. And this shows the viremia of the vaccinated animals. So they first get viremia from the vaccine, and then when they get infected, reinfected, it's a heterologous virus, and they show again a viremia curve. And of course, you already see that there is some protection to PRRS, because the vaccinated pigs have lower viremia than the non-vaccinated pigs. But maybe we can use this, measure this on animals, and use that as an indicator trait for viremia in an actual infection when the animal sees it for the first time. So if you look at, and then again we look at viral area under the curve. If you look at the vaccinated animals and you plot the area under the curve that they have post-co-infection against the viral load they have pre-infection, there is a positive relationship, but it's not very strong, correlation of 0.27. So there is a tendency for pigs that have higher viremia following vaccination to also have higher viremia following subsequent co-infection with PRRS and PCV2. But that's not really what we're after. We're really after, you know, do pigs that have higher vaccination or higher viremia after vaccination, do they also tend to have higher viremia when they have an actual non-modified live virus infection? So we really want to compare the dark lines here against the light blue lines here. And we can do that by estimating what's called a genetic correlation. Okay, and this is the genetic correlation of 0.93 here, is the genetic correlation between the genetic correlation between viral load after vaccination with the viral load after co-infection without vaccination. And the way you can think about that is, you know, if there are boars that have progeny that were vaccinated, okay, so on those progeny we have their vaccine response. Those same boars would also have had progeny that were in the non-vaccinated group, okay, and so those non-vaccinated animals have a viremia curve, a viral load when they from the co-infection when they see the PRRS for the first time, and that allows us to estimate. The connection is genetics, the common sire, and that allows us to estimate that genetic correlation. And the 0.93 here says that if you select animals that have high viral load after vaccination, or with low viral load after vaccination, you will tend to select animals that also, after a real infection, will be more resistant. Okay? Now, note here the standard error, and I made it small so you don't see it very well, but it's a huge standard error. And that shows one of the problems of these genetic studies. Even though this represents data, and Janelle Dunkelberger was part of her thesis, this is based on 400 animals. It's nearly not enough to get accurate estimates of genetic parameters. Okay, so this is promising, but we need more data. We need to infect more pigs, Bob, to really get to the bottom of this. Okay? But it's a promising result, and there's some other indications that you know, antibody levels after vaccination may be indicators of response to a real infection. So indicator traits, and of course in the study that Graham talked about, we're measuring lots of potential indicator traits. Okay? And this study really will allow us to delve very deeply into lots of possible indicators that can predict resilience to a multitude of diseases, a combination of diseases. Okay. Now, a very good example of this in dairy cattle, of an indicator trait, is work that was done by Bonnie Mollard at the University of Guelph, where she measures cell-mediated response and antibody-mediated response using fairly simple tests that can be done in a clean environment. And they're doing that on dairy bulls at a young age. Okay? So they measure two measures of, and they're quite heritable. And then she's shown that that animals that have low, a combination of low cell-mediated immune response and low antibody-mediated response, they have higher disease incidence. Okay? And it's a multitude of diseases. And this is actually being used by CMAX, which is the dairy genetics industry in Canada, as a proprietary test to market their bulls that have improved immune response. The same technology she is now using in the project that Graham talked about as a potential biomarker. Okay? Too early to say whether it's going to be useful also in pigs, but it's one example where there is good evidence in another species where these types of biomarkers measured on healthy animals in a clean environment can tell you, give you information about how robust animals are to disease and to a multitude of disease. Okay, so that's the first part, so integrated traits, ideally ones that we can measure in the nucleus. The next step is, well, maybe we can collect things at the commercial level, okay, and then utilize that for selection in the nucleus. And that's being done actually by some breeding companies, where they measure reproductive data on in the multiplication phase, where there may or may not be disease, but you could also measure phenotypes on the crossbred market pigs, and again you could target phenotypes from pigs that were exposed to disease. Okay? And then utilize that. These are all offspring of the animals in the nucleus, right? So you can utilize this data to estimate breeding values of animals up in the nucleus. Okay, so the potential is there, and it's being done. The challenge is that if we don't have pedigree, we won't be able to utilize that. We need to know. We have to have individual pig data, and we need to know who the parents or grandparents of these individuals are. Otherwise, we can't use that information to estimate the breeding value. So, and often we don't have that pedigree, and especially with using mixed semen, you can't determine the pedigree. You lose track of the pedigree. And there's time delays, and that increases the generation interval. I don't know why some of my symbols changed here, but there should be an arrow going up. So increasing the generation interval, and we want to spin these generations as quickly as possible to speed up the genetic gain. So that's an opportunity, and you could target, again, phenotypes collected under disease. And there could be growth rate under disease, which is really a measure of resilience, right? And you could use that in selection of the pure lines. Another potential solution there is genomics, using genomics. And this goes to genomic selection, genomic prediction, and genomic selection. And even if you don't have clean or markers that have large effects, but if it's a trait that's affected by many genes, you can use this technology that's called genomic prediction and genomic selection, which involves creating a training population, a group of animals where you measure phenotypes, and you genotype them for a large number of markers across the genome. And we can now do that. It's still not cheap, but it's a lot cheaper than it used to be. We can genotype an animal for less than $50 for tens of thousands of genetic markers across the genome. And that's enough to do this genomic prediction and genomic selection. And it's been implemented in dairy cattle, and is also being implemented and has been implemented in several pig breeding programs. So you use this training population to estimate the effect of every SNP, every genetic marker. So for every genetic marker, if there are 3 genotypes, AA, AB, and BB, for every genetic marker, you get an estimate of whether the AA increases the phenotype versus AB versus BB. Okay, and you get that for every marker, not just for the markers that are significant, like WR, but for all the markers. And then by treating them statistically in an appropriate way, you can utilize information for prediction. So the idea then is that you go to your selection candidates, the next generation animals you want to select, and you genotype them for those same markers, and you then apply these estimates to those genotypes, and then you get what's called a genomic prediction. Okay, so based on their genotypes, If they have a higher proportion of favorable genotypes, they will have a higher genomic prediction, and they're estimated to be genetically better. And you can use that for selection. Okay, and that's been done. A lot of the training data is generated within the nucleus. Okay, so you can feed intake, for example, is expensive to measure. With this technology, you can measure phenotype. You still have to measure it on a substantial number of animals, but you can use genomic prediction to leverage that phenotypic data to other animals that are not phenotyped. But you can also use that for data that you may be recording in the field. So the idea then would be that you would, for example, you take a group of market pigs, and when I say a group, I'm talking about probably a couple of thousand market pigs. You genotype them using these SNP panels. You get their phenotypes, which might be growth rate under disease, measuring resilience. And you use that as the training population. You use that to estimate the effect of every SNP on this panel on growth rate under disease. Okay? And then once you have those SNP effects, you can apply that in the nucleus. So now you can genotype animals in your selection candidates, genotype them for the same markers, and apply these SNP effects, which now reflect the effect of those SNPs on growth under disease, and get genomic predictions that you then use to select animals for. Okay? Now, one of the advantages of this is that it doesn't require pedigree to be tracked through the system, which is one of the problems if you don't have genomics, is that you need to use this data up here, you need to follow the pedigree. But with genomics, you don't need that. Okay? All you need is the genotypes here, and you can estimate the effects of every SNP, and then utilize that in the nucleus. So that's another strategy that can be used. And then again, yeah, so it's the phenotype ABV markers or genomic prediction. So finally, you could also, rather than going to the field, you could collect data in special experimental facilities, like we did in the PHGC, right? And like what we're doing now in the Genome Canada project. So the idea there would be that you have your nucleus animals and you have the experimental facility where you do a disease challenge. And ideally what you would want to do is you would measure, you would send relatives of your selection candidates. So part of the litter you would send to the disease challenge, for example. And Highline, a layer breeding company in Iowa, has done that very effectively for many years already to breed chickens that are more resistant to Marek's disease. And Marek's disease is one of these diseases where the pathogen, the virus, mutates a lot. And so every year they take the most virulent strain of the virus and they test progeny of their roosters in this disease challenge facility, and they then select roosters based on the % of the progeny that died following this challenge. And that's been fairly effective. It's one of the reasons why the livability of the survival of Highline chickens is better than, on average, than other companies. And you know, in principle, we could do the same thing in pigs. It's expensive. But again, genomics may be the solution, may be a solution, because then you don't have to necessarily measure this on close relatives, but you measure that on enough animals from those lines such that you can estimate the effects of SNPs on the panel on the ability to resist that challenge or survival to that following challenge. And that's what we can do already based on the PHGC. And we actually tried that. Where we use the PHGC data as a training data, and then use that to, you could potentially use that to select within the nucleus. And we tested that. So remember, we have the NVSL trials and we have the KSO6 trials. And this is work that Emily Wade, another graduate student, did, who now works for Genus ABS. What she did is she did it both ways. So she estimated the SNP effects based on the NVSL data, and then see whether she could predict the KSO6 data. So training based on viral load following an NVSL infection, can that predict the viral load following a KSO6 infection? So this also does another thing. It also tested whether we can predict resistance to heterologous strains, because these are about 89% identical at GP5. And she also did it the other way around, so train on KSO6 and predict NVSL. What she found, so the first one where she trains on KSO6 and then predicts NVSL, when she used all the markers across the genome, the accuracy of the genomic prediction was about 0.45, which is actually reasonably good. You'd like it to be better, but given that these are not always the same genetics, it's not bad. She also looked at, well, what if I just use WR, right? Ignore the rest of the genome. And when she used just WR, okay, the green one, she got about half of that. Okay, so actually a little over half of this prediction comes from WR. And that just reflects that WR has a sizable and a predictable effect on viremia, even across heterologous strains. And then the rest of the genome, when she looked at how predictive is the rest of the genome, excluding WR, there's some predictive ability, but it's not very good. And that, again, reflects that it's many genes with small effects, and those small effects are very difficult to predict. But, there's some predictive power there. When she did it the other way around, so training on NVSL and predicting KSO6, overall their accuracies were a bit lower, but still the same trend. So using the whole genome is best. Using WR, you get over half of that, and the rest of the genome has some predictive ability, but not all that much. Okay? So I mean, it's some encouraging results, but still not where we really want to be. In terms of, but it would be, certainly WR has the potential to, can be used to improve resistance, and then we've seen also tolerance and perhaps infectivity or rebound. And the rest of the genome can add some predictive ability also. There's a poster this evening, Janelle Dunkelberger, where she did this using the vaccine response, and then either PRRS-only infection or co-infection. And so go look at her poster. And what she found is that also it's primarily WR that gives you the predictive ability. Okay, so to summarize, so resistance, host response to PRRS, and also this also holds for many other infectious diseases, has a sizable host genetic component. But unfortunately, it's not simple. They're complex diseases, many genes involved. Current breeding programs have limited opportunities to select for improved health because we don't really have, at the moment, we haven't had measures that we could take on the selection candidates or their close relatives. But there are things we can do. One is indicator traits, and we're hard at work to develop those. We've seen some information already from the PhDC trials that Joan talked about, and then of course the work that that Graham talked about. We could look at collecting data in the field, okay, and we're working on with some of the companies' breeding programs to accomplish that. So feeding that back either through genomics, feeding this information back into the nucleus. Regardless, it's going to require continuous recording of disease-related traits. And so there's still a lot of work needed to determine what are the best traits to measure. And in order for them to be useful for genetics, they have to be measured. You have to be able to measure them on a large number of individuals, and that's where we need the help from everybody: genetics, the veterinary side, and you saw this also this morning. There's lots of data that's being collected in outbreaks. A challenge for us to use that is that usually it's not on individual pigs that are identified. But ideally, you would measure that on individual pigs and then also get a hair sample or a blood sample for DNA. And then we can work with that. And genomics definitely has the potential to help the use of all this information for selection in the breeding programs, either individual markers that have large effects or many markers across the genome using genomic prediction. Okay, with that, I want to thank all the partners again, and thank you for your attention, and I'll turn it back over to Bob.